Hello, I am Kaustuv Sanyal. I am a professor and chairman of Molecular Biology and Genetics Unit at Jawaharlal Nehru Centre for Advanced Scientific Research in Bangalore, India. I have obtained my master's degree from Madurai Kamraj University, and then I did my PhD in yeast genetics at both Institute in Kolkata, before I went for my postdoctoral work at the University of California in Santa Barbara. I joined here at JNCASR in 2005 as an assistant professor, and my research interests include molecular understanding of the process of chromosome segregation, and I use various pathogenic budding yeasts as my model. So I started working on several proteins that are required for segregation of chromosomes during mitosis, which is the process that helps one cell to divide to give rise to two daughter cells. During my postdoctoral studies, I got interested in an organism called Candida albicans that causes various diseases in humans, especially those who are immunocompromised. At that time, with Professor John Carbon at University of California, Santa Barbara, we identified an important chromosomal DNA sequence, which is called the centromere. Centromere assembles several proteins that form the kinetochore, which is the binding site of spindle microtubules that facilitates the process of chromosome segregation. So once we identified the centromeres of Candida albicans, we realized that unlike most organisms where the DNA sequence is conserved across centromeres in a given organism, in this case each centromere sequence is unique and different. When I started my own lab in JNCASR here in Bangalore, we got interested in understanding the centromere evolution by studying several pathogenic budding yeasts which are related to Candida albicans. While doing so, we realized the centromeres are the most evolving DNA sequences in the genome. That led to an interesting understanding that whether the centromere DNA sequences are the driving forces of speciation. So I have started the pathogenic fungal biology research at the molecular level in India, with a major focus on the fungal centromere biology. Though you know that centromere is a chromosomal locus that serves as a binding platform for mitotic spindle. So during in the initial phase of my research in India, we identified and analyzed centromere sequences from many major pathogenic fungal lineages, and we realized that centromere DNA sequences are evolving quite rapidly. The centromere DNA sequences can be quite different from each other even in the same species. That helped me to realize that there must be factors other than the DNA sequence that determine where the centromere should be formed. To test this hypothesis, we deleted a native centromere in Candida albicans, and we found that a region next to the native centromere which we deleted formed new centromere, which we termed as neocentromere. This suggested that centromere identity is not determined by the DNA sequence alone, but the spatial location of the region of a chromosome in fact determines where the centromere should be formed. We have performed more research in understanding the 3D conformation of the chromosomes using techniques such as chromosome conformation capture to understand the spatiotemporal regulation of centromere identity. We perform various collaborative research from scientists across disciplines such as mathematicians, physicists, and chemists. So one of such collaborative ideas came when a scientist named Christophe was visiting India to attend a meeting in Delhi. We were casually discussing about exploring a library of Candida albicans. While talking to Christophe, we discussed about possibility of utilizing Candida albicans overexpression ORF-OM library that he has created with another collaborator. We were interested in understanding how what are the determinants of chromosome segregation, and this library we thought would benefit us to screen for factors that may regulate chromosome stability. So he immediately agreed to collaborate on this project, and we started writing up a proposal. So we had several discussions over Skype, and we came up with a proposal and submitted to CEFIPRA. And we were lucky that the project was funded and we started our collaboration. So one of the advantages of having a CEFIPRA project is the exchange visit. So I could go and visit Pasteur Institute in Paris, where my partner Christophe works. The French PI lab was really well equipped with various high throughput techniques. I learned various facilities that they have, and I could also interact with many other faculty members of the unit. I could exchange my research ideas and learned a lot. In fact, Christophe also visited our lab twice and interacted with my students and postdocs here, and it was a very enriching experience for both of us. Eventually, when the project got started and was moving well, my PhD student who was involved in the project had to learn how to do high throughput assays and high throughput screens. So she went to the Pasteur Institute in Paris to learn the techniques because we were not aware of such techniques, and she spent about 3 months there. Thanks to CEFIPRA again, that made it possible, and she came back and we developed the same systems and same facility right here in Bangalore. So by doing so, we screened about 1000 genes of Candida albicans, and we analyzed what is the effect of overexpression of each of these 1000 genes on chromosome stability, and finally identified one of the factors which is named as CSS6, that is present only in Candida albicans and some other related pathogenic yeasts. The gene, the new regulator of genome stability that we found in Candida albicans and named as CSS6 is an interesting protein, because what we realize that when we overexpress this protein, then the cells are arrested at metaphase stage of the cell cycle, and they cannot proceed to the next stage of the cell cycle. And when we depleted the protein, and we found that this protein is essential for viability of Candida albicans cells, because they were again arrested and in the cell cycle. But interestingly, unlike the metaphase arrest that we observed when we overexpressed the protein, when we depleted it, we found that the cells are arrested at the anaphase stage of the cell cycle, which means that these cells with depleted levels of CSS6 could not proceed to mitotic exit. So, in other words, what we have shown that we identified a unique protein that is present only in Candida albicans and related pathogenic yeasts and not present in humans, is so finely regulated that its abundance or its depletion can cause cell cycle arrest and thus death of Candida albicans. So this raises a possibility of targeting CSS6 as an antifungal drug target, which since it doesn't have a human homologue, so if we can identify a specific inhibitor of CSS6 function that can specifically stop cell division of Candida albicans without much effect on humans. As I was mentioning, other than CSS6, we identified also 5 different proteins such as CLIF4, KIP2, BFA1, AAC1 and MCM7. While most of these genes have not been characterized in Candida albicans, but their homologues were known in Saccharomyces cerevisiae. So as I was mentioning, by screening about 1000 genes and their effects on overexpression on Candida albicans cell cycle, we identified 5 factors other than CSS6, and they are MCM7, CLIF4, KIP2, BFA1 and AAC1. Now these proteins, the function of each of these proteins are not characterized in Candida albicans, but their homologues are identified in Saccharomyces cerevisiae and they were quite well characterized. And these proteins, as I mentioned, they are associated at various stages of the cell cycle in Saccharomyces cerevisiae. We focused on CSS6 because the homologue of this protein was absent in Saccharomyces cerevisiae as well as in humans, and as we have shown that CSS6 overexpression or repression can cause cell cycle arrest of Candida albicans. Since both fungi and us are eukaryotes, it is difficult to find a target or a protein that can be inactivated specifically to the human pathogens without any effects on humans. Since CSS6 is present only in Candida albicans and its related yeast species and not present in humans, this can be used as an attractive target for developing antifungals. So Satya, you are also working with another pathogenic yeast in the lab, which is Cryptococcus. Can we talk a little bit about Cryptococcus cell cycle, its genome, what are the important features of Cryptococcus?
So, unlike Saccharomyces cerevisiae, like in this model organism, it's interesting to see that the aspects of kinetochore dynamics are diverged. All throughout the cell cycle in cerevisiae, Cryptococcus, the kinetochores are clustered, but in case of Cryptococcus, they transition from an unclustered situation to a clustered situation in mitosis. So it is important to look what controls this dynamics and what is the functional implication of clustering and unclustering of kinetochores, because it's a pathogenically important organism, because it infects immunocompromised individuals like Candida albicans. So looking at these interactors would be benefit to identify a antifungal drug that perturbs kinetochore dynamics.
Okay, Priya, you were working on Candida albicans, and you are looking at morphological transition regulators in Candida albicans. Can you talk a little bit about it, because those morphological transition factors may influence the virulence properties of Candida albicans.
Yeah, so one of the major virulence factors for albicans to propagate is basically how it can transition from a yeast, which is around free floating cells, and then it can go to a very elongated cell like morphology, which is hypha. So also it has different other properties, like it has to adhere to the epithelial surfaces, and then only it does the infectious properties. So there are certain genes known which already have been characterized in albicans and can be used as antifungal drugs, but through this screening process that I have done, it is also possible that we may find new novel regulators which were not identified earlier and are actually not correlated to any of the human genes. So yeah.
We work on pathogenic fungi, and we are interested in understanding how chromosome segregation happens in these organisms. For example, we work on a pathogenic organism called Cryptococcus, which primarily infects immunocompromised individuals, and we are interested in understanding how chromosome segregation happens here, because it was reported that by the means of aneuploidy mechanisms, it has been shown that it acquires resistance to azoles. So what happens here is that, say for example in Cryptococcus, we have identified, and several other groups have reported bioinformatically that various kinetochore protein components are missing, and moreover this organism shows a differential kinetochore dynamics. For example, during G1 state it shows an unclustered kinetochore signal, whereas in G2 stage it shows a clustered kinetochore signals. So we wanted to see why there is a transition between a unclustered kinetochore versus a clustered kinetochore signals. So that we are interested to study that, say for example, if we perturb any of the kinetochore components, is the cell cycle progression is affected or not, and through these means can we develop any drugs that can tackle Cryptococcus. On the other half, we are also interested to look at to see whether there are any genome-wide changes, because from a stage where you see unclustered kinetochores and to a transition where there is a clustered kinetochores, there is also the changes in the genome dynamics. So right now we are also interested in looking at how the genome changes during cell cycle process in this organism.
Long live India and France friendship.